two-dimensional space, some new ways that we've been thinking about presenting the data, complex data sets.
So welcome everyone, I'm Federico, I'm a postdoc in the lab. And so the main aim of this presentation will be not to show you a ton of data, but to hopefully really show you the ideas that go behind the development of the methods that we have developed in the lab, and especially to show you what are some of the most fundamental basic computational issues that you're going to face when you analyze this kind of data, and to show you how this essential issue can be solved in different ways, and this leads to different methods. So essentially to show you what is the common thread that links a lot of this method behind the scenes. And I know that computational stuff can be pretty dry, and I want this to be as clear as possible. So please, 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 if there is something that you missed during the presentation, don't wait till the end, but interrupt me immediately so that we can get the issue clarified without going further and then losing track of everything. Also, I mean, some of the stuff, especially this, there is a little bit of spade stuff here, which has already been touched upon by many of my lab mates. And so, I mean, this is the chance to really go in detail on some of the aspects. So if you have issues about spade or something that you want clarified, both me and also Rob afterwards will give you a hands-on demonstration. So this is the place to ask such questions. I will start with a pretty obvious slides, which, I mean, I'm sure everyone is familiar with gating, and you have been doing gating on your fluorescent data. And I'm sure that the talks that we had yesterday and this morning have already convinced you that this approach is not really very well suited to the analysis of very multi-parameter data sets, because the number of dimensions increases a lot. But I mean, this is it's important that we know where we come from, which is essentially gating. And people have already shown these slides a number of times, but I want to put a little different spin on it. So here you see that, of course, if you have 32 parameters, you have like almost 500 biaxial plot to look at, which is one way of putting things, which is certainly true and scares you away from looking at all these plots. But there's also another issue, which is that if you imagine each cell as a point, then these cells really live in a 42-dimensional space. So it's your whole data set is a gigantic cloud of points in a 32-dimensional data sets, okay? So this would be, if it's only 3-dimensional, of course we cannot visualize 32-dimensional, but you have to imagine this gigantic cloud that is residing in this high-dimensional space. And of course, each biaxial here is just a projection of this cloud on 2 specific dimensions. But the point I want to make is that even if you know where your cell is in this gigantic multidimensional space, it might not be trivial to find the sequence of biaxial plot that will lead you to that cell, okay? So it's not just a problem of visualization, but it's also a problem of finding what is the series of projections in 2 dimensions that lead you to that specific point in high-dimensional space. And so that's the reason why we need some of the tools that I'm going to show you in a few minutes. And also, Kara has already shown these slides. These slides that show you that these points, as I was telling you have to imagine this gigantic cloud of points, which here is represented only in 2 dimensions. And of course, this cloud is a continuum. So it's not very easy to draw boundaries here. I mean, where do you define the boundaries in this continuum? It's very hard to tell, right? And so these problems become all but magnified when you're dealing with 40 dimensions instead of 4 or 5 or 6. So the main let's say, coordinates of the problem. So the main raw data with which you will work when you're dealing with a set of experiments are going to be the number of cells. So you're going to have like hundreds of thousands or millions of cells per sample. You're going to have 40 markers, which are give you a gigantic multidimensional space. And then, as Eli has shown you, there are ways that we now have to barcode the samples together so we can also run a ton of samples, so ton of sample, ton of cell, ton of markers. And the end result, and this is something that Wendy has already mentioned, it's that you're going to spend way more time analyzing the data that run in the experiment, okay? And not only, I mean, she was arguing about the reagents development, which is definitely true, but I would say that probably you're going to spend more time analyzing the data than the reagent development and running the experiment combined. So making, like if you really want to mine this data extensively, you can go crazy and really spend a lot of time. And I compiled here a very brief list of the things you normally would want to do. Of course, I mean, you can always find experiments that don't fit this particular scheme, but let's assume the most basic side of experiment that you will want to do. So the first thing you will want to do probably is to have an idea of the structure of your data so that you can find out where your population of interest is or are, if there are more than one. So the first issue is identifying the populations. Then you probably want to compare multiple individuals, like multiple patients or patients versus healthy, or you might want to compare multiple stimulations. So what happens if I give myself IL-7 or this other cytokine or this other stimuli or this other condition? So this is another basic question you probably are going to have when you do your experiment. And the last thing, not the last thing, but another thing you will want to do would be to make statistical tests for your hypothesis. Okay, so I think that this population, this feature of my data set is somehow correlated with remission or progression of disease or whatever, and you will want to make statistical tests about this hypothesis. So a big disclaimer is that everything I'm going to present you now is only going to deal with these first 2 points. So this method I'm going to present you are going to allow you to visualize the data and compare the condition, but they're not going to allow you to do tests on your hypothesis. For that, Rob, after me, has developed a method that allows you to do exactly this, and so it will detail the workings of this method after me. But for now, for the purpose of the stuff I'm going to show you, I'm just going to be focusing on the first two parts, okay? And another thing that I want to clarify is this definition that we often use in the lab of functional versus phenotypic markers. So the phenotypic markers are usually your CD molecules, CD3, CD4, CD20, CD19, and this is the stuff that define the cell populations you're looking at. And usually, I mean, for the sake of simplicity, as I was telling you, you can always find counter-arguments, but for the sake of simplicity, let's assume that these are more or less static in your experiment. So this is more or less constant. You're looking at this set of surface markers because you're interested in this set of cells, okay? And then what we call functional markers, which are often dynamic, so it's the stuff that is changing over the course of your experiment or between health and disease, is or your phosphomarkers, the readouts of the inner state of the cell, okay? Of course, I mean, cell populations can vary in frequency between health and disease, so the same population can be expanded or contracted in different patients and all of that. But we more or less, again, for the sake of simplicity, let's assume that this is more or less static, and this is what changes across the different stimuli. And another, why this distinction is important also has to do with this concept that people have already shown, but on which I want to go into more detail, which is the concept of building the structure of your visualization and then overlaying information on top of the structure. So you build the general framework, and then you want to annotate this framework which features that are interest for you and for understanding your data. And usually, you will build the structure, like the general organization of your data on the phenotypic markers, and then on top of that, you will overlay, so you will annotate this structure with the functional markers, which are dynamic in the course of your experiment. So all of the things that I'm going to discuss today start with this concept of clustering, on which I want to be extremely clear up front. So the result of your experiment from the point of view of the bioinformatics is essentially a gigantic table where each cell is a row and each protein that you've measured is a column. And the numbers represent the expression of protein A in cell 1, protein B in cell 1, or protein A in cell 2, and so on and so forth. And so clustering essentially deals with identifying which rows of these metrics are similar, so which cells are similar. And if we go back to the example in our high-dimensional space, essentially this means that if these are our cells in a three-dimensional space, then we identify that this group of cells here are similar because they sit together. This group is similar because they're close together. So it's essentially finding lumps of cells that are similar and live together. Okay, so this is so you end up with a bag of clusters. And the thing I want to be very clear is that clustering is a very fundamental difference with gating. Because when you do gating, you put your prior knowledge in identifying a subset of cells. So by definition, at least according to your intuition, the stuff that you are gating out is a population, is a distinct population, because you are putting your prior knowledge and you're calling that a population. When you do gating, well, sorry, this is when you do gating. When you do clustering, then the thing is different. So what we are saying here is that this group of cells are similar in this cluster, this group of cells are similar in this cluster, this group of cells, but this does not mean that the cluster themselves might be similar as well. So I'm not saying that each cluster is a population, because a single population like CD40 cell might be split among different clusters. The only thing I'm saying is that the stuff that falls in the same cluster is similar to each other. Okay, so I hope this is very clear. We're not saying that each different cluster is a different population according to your biological intuition. We're just saying that the stuff that's in the cluster is similar. Okay? Everyone cool with that? Okay. So once you have this gigantic bags of cluster, then of course the first question that you want to ask is how you organize them. And the most basic way to organize them is essentially to connect each cluster to its most similar cluster in the data set. Okay? So you essentially end up with this structure that is called a minimum spanning tree, which essentially means that it's the number of connection that minimizes the difference between each cluster. So if you give a score to the difference between each pair of clusters, okay, you give a score to the difference between this and this, a score to this and this, etc. This structure is the structure that minimizes the difference in the whole data set, okay? So there is no, like, this is the minimum distance that you can use to link together all your clusters in your data set, okay? And this structure is called a minimum spanning tree. And this idea is what is at the base of SPADE, which, not surprising, because spanning tree is actually in the title. So this is the way to organize the cluster that is at the basis of SPADE. So connecting clusters so that the sum of distances is minimized, okay? And the way that SPADE works a little bit more in detail, so there is first of all, there is a step that is called density downsampling that has to do with that. So if you take your cytometry, like your flow cytometry data set, you usually end up with regions of your space that contain a lot of points and regions of your space that contain fewer cells, okay? So you can see in this plot here, I'm sure you're familiar with this density plot, but essentially the fact that here there's a strong yellow color means that there is a lot of cells in here and not as many cells in here. So the first thing that SPADE does is to normalize all this away so that essentially your data set looks like it has the same density in all the region of the space, okay? So this is essentially the same thing. So the same region of the space is occupied by your data set, but the density has been evened out across all your data set, okay? So the number of cells, like here, the number of cells was way greater than the number of cells here. After the transformation, the number of cells here is equal to the number of cells here, okay? And the reason why SPADE does this is to give a higher chance to very small populations to be identified, not to be swallowed up by some other cluster. So after that, you cluster the data, and this is something which I hope I've already explained pretty well. And then you do the minimum spanning tree. So essentially, you take your clusters and you connect them so that the sum of distances is minimized across all your data set. And then, of course, you want to, I mean, from now on, it's just technical details, but there needs to be a way for you to go back to the original data, which has this density, which is higher. So essentially, the thing that you're doing that is called upsampling is just taking your clusters here and assigning each point in the original data set to its nearest cluster, so that at the end of your analysis, all the data that was here has been assigned to something. Okay, so essentially you're saying this cluster here lives in this point here, so I will assign all the cells of my regional data set to that clusters over there. And this is just, I mean, it's I would call it a technical detail. It's not a fundamental difference. It's just so that every cell in your data set has a place in your SPADE analysis. Okay, and once you've done upsampling, this has to do with the way that you then color the tree and annotate the tree to have additional information. So of course, I mean, you can then make the circles bigger or larger according to the number of cells that you have in that cluster. You can color the cluster by marker expression, by whatever stuff you want to annotate your data set with. So I'm sure that you have seen these tree hundreds of times, but so there are only two very important points that I want to make about this tree. So three important points. So point one is that, once again, we're not saying that each cluster here is a different population, okay? So the reason why there are these bubbles around the cluster is because someone essentially determined that all these clusters are essentially representing the same immunological population, and so we have grouped them together, okay? So this goes back to the issue that I was telling you about before. We are only saying that stuff in the same cluster is similar. We're not saying that two different clusters are two different populations according to your immunological knowledge. So this is the first point. The second point is that the way that you arrive at this definition is because someone has gone through all the trouble of visualizing marker expression on this tree and then manually calling that, okay, since these are CD3 positive, then there must be T cells. These are CD8 positive, so these are your CD8 T cells. These are CD4 positive, so these are CD4 T cells. So essentially, the output of your SPADE analysis won't be this tree with all the bubble and all the information telling you this is T cell, this is B cell, but it's going to be a tree on which then you can project information and annotate it so that you end up with this definition. And the other point that I think David asked about yesterday is that the only, and I'm going to go back to this later, but the only thing that is meaningful in this tree is the connection between the nodes, not the position of the nodes, okay? So if I take this node here and I move it all the way down to here, or I take this bubble here and I move the nodes away here, it's always going to be the same SPADE tree. It's always the same SPADE tree because the position of the node doesn't have any information. The only information is the connectivity of the node. So who is connected to whom, okay? So the other thing that this slide has been shown already, but the other thing that I want to once again emphasize is that the good thing about this visualization is that now you have a reference structure which you can annotate with your information. So you can overlay additional information on top of this structure. And like, for instance, here, what you see here is phosphostat-5 activation, the basal activation. And this is after IL-7 stimulation. And of course, I mean, there are multiple ways to show this data. So this is like the total amount of phosphostat-5. But if you want to do a more precise analysis, you can essentially normalize over basal like you would do normally. And so essentially, you're taking the full activation of STAT5 over basal. And so you subtract basal from the activation. And this shows you more clearly that this is the population that is responding to phosphostat-5. OK? So up to now, we have been talking about essentially taking one sample, analyzing, and showing marker expression across different stimulation. But one important issue is what you do to analyze multiple samples, okay? So, and I want to stress this because it's the inspiration for something else that we've done later. But the point is that when you do a SPADE analysis, the way to analyze multiple samples is that you pool all your data together and you make a single SPADE tree of all your data, okay? So in the end, what's going to happen is that different samples are possibly going to populate different regions of the tree. Like, for instance, the first sample here lives in this section of the tree. The second sample lives in this other section of the tree. But the important point is that since this tree has been built with all the data, the stuff that falls into this specific cluster here can come from any number of samples, right? So the cells that are in the cluster can come from the healthy, from the disease, from whatever. They can come from any of your samples, right? So the only thing that is distinguishing the samples is how they populate the tree, okay? So for instance, this cluster here will contain maybe only cells from the healthy patient and no cells from the disease because here is smaller. But there are clusters here in the middle that will contain cells from both the healthy and the disease because the structure and the clustering has been done with all the samples together. Okay, so that's all I have to do with SPADE. Just the other two things that I wanted to tell you is that SPADE is essentially available in two formats. One is as a Cytoscape plugin. So Cytoscape is a bioinformatic open access tool for the visualization of network and has been adapted for using SPADE. So essentially, you can take Cytobank and use SPADE as a plugin and do your SPADE analysis. Sorry, not Cytobank. I always get confused. Cytoscape. So Spade has been implemented as a Cytoscape plugin. And also there is the Spade version of Cytobank, which Rob is gonna show you later. And it's another way that you can access Spade. So what are some of the issues with Spade? So the first and most important issue, which is often overlooked, is that the tree structure is extremely sensitive to minor variations in your data. So essentially, in that tree, changing just a couple of connections can result in a tree that is completely different, okay? Which means that essentially, every time you run your Spade analysis, you're going to get a different tree. So that's very important. If you take the bone marrow data set from Sean's paper and you run it, you're not going to get the same Spade tree, because the analysis is stochastic and it's sensitive to very minor variations. The other thing is that you need to
It's important to say, though, that it doesn't mean that the data is
Oh,
different
no.
as it's represented. It just means that when you, I mean, think of it this way. If you have 3 dimensions in a sphere and you take away 2 dimensions, you've got a point. So imagine what happens if you've got a 40-dimensional object and you're taking away, 37 dimensions. If you look at that object just slightly differently in high D space, it looks different when it's projected down to 2D, but it isn't any different in terms of its original structure.
Oh, yeah.
And as useful as Spade has been for us over the years, last 3 or 4 years, it's become more and more difficult for us to accept that it doesn't need fixing. So that's why we've been moving towards a Spade 2.0 version that is not varying from one experiment to the next, and that's one of the things that I think you'll get to.
Yeah. Mm-hmm.
One other thing just to add on to Gary's comment, even though the trees are technically different, they still look very similar. So the markers that you use to cluster on will give you a certain shape of your tree. So I've done, like, for normal bone marrow, I've done a series where I'll take a certain set of markers and I'll run 3 Spade analyses, and then I'll run 3 more with a slightly different set of markers. And the general shape of the tree is almost always the same for a given set of markers. So when I do normal bone marrow, B cells are always next to monocytes because they both have high HLA-DR, and so they always end up sort of as 2 different branches on the same side of the tree. So it is stochastic, but it's not like you get something that's totally different. And the progression, B cells always go from stem cells out to pre-B1, pre-B2, mature B cells. Like, I've never had a tree that didn't follow that pattern if I used the standard markers. So it's a little different, but there's still a very strong underlying similarity there.
Yeah, so the overall, like the qualitative conclusion you can get from the tree are the same because the data is the same, but I mean, as we discussed, the details of the construction of the tree are different. And so the other thing is that you need to cluster all the samples together when you do your Spade analysis. And this means that if you want to add a new sample to your analysis, then you have to essentially rerun your clustering and you rerun your Spade analysis from scratch. Okay? And the other thing which is very important, and I mean, this is not a limitation, but it's just a way that Spade is defined, that it will always give you a tree. Okay? So the fact that you see a tree doesn't mean that there is some sort of very beautiful organization in your data, because even if you use random data, it will give you a tree, because that's the way the algorithm works. That's an important thing to appreciate. So this is not a limitation, but it's just the way that the algorithm works. It will always give you a tree, even if you use random data. Okay? Then, of course, the tree doesn't mean anything, but that's up to you to get at that point. And the other thing that I already told you is that in a Spade tree, and this is very important, only the connections are informative, not the position of the nodes. You can shuffle the nodes around, like drag and drop. You can do that on the Cytobank interface, and it will always be the same Spade tree, because only the connection between the nodes are what's important in the Spade tree, not the position of the nodes in the page. Okay. So one thing you can do, so this is essentially your very simplified Spade tree. So one thing you can do to sort of going beyond this limitation is to augment this structure by introducing connection, not just connecting each cluster to the most similar ones, but also connecting cluster to stuff that is more different. Okay. So instead of connecting only the stuff that is very similar, you start comparing all the clusters with each other, and you add connection to represent this comparison you're making. And of course, I mean, at a certain point, you have to, like, you don't want to connect everything with everything because otherwise you don't understand anything anymore. So you might want to set a threshold in similarity after which you don't have connection anymore. But as a general framework, you can imagine that SPADE is just connecting together the stuff that is most similar, but you can also go beyond that and connect together stuff that is more diverse, okay? And so essentially, more in general, you can represent single cell data as a graph, okay? And a graph is a mathematical object, where each node of the graph is a cluster of cells, and the edges, so the connection between the nodes, have a weight associated to them, which is proportional to the similarity, okay? So if these two clusters are very similar, they will have an edge with a very strong weight. If they're very different, they will have an edge with a very low weight, okay? And as I was telling you, the minimum spanning tree is just a special kind of graph, so we're not inventing anything new here. We're just saying that the minimum spanning tree is a special kind of graph, but we can make a more general graph that represents this data. And I mean, there are two very important properties for the purpose of this stuff that the graph has, and these are the connectivity and the layout. So the connectivity just tells you who is connected with what, okay? So the edges in your graph. But then the other important property is the layout. So how you put the nodes on the page. Where do you, on a page, where do you put node 1 and node 2 and node 3, okay? And these are two distinct properties. And actually, the layout can give you very important visual cues, because these two graphs here, they are exactly identically the same graph. So these are the same nodes with the same edges with the same weight. The only difference between this representation here and this representation here is the way that the layout has been constructed, so the way that the nodes have been placed on the page. But the graphs are exactly identical. There is no difference between these two graphs. And so this is the Eli in the lab started using this concept of force-directed layout, which is essentially a way to lay out the graph in the page so that the stuff that is similar to each other ends up in a similar position on the page. So you can imagine that each one of these edges acts as a spring, and the stronger the spring, the more close the nodes end up being on the layout, okay? So this layout here is already telling us that this group of cells here are very similar, these groups of cells here are very similar, these two cells here, these two clusters are very different from all the rest, okay? So thanks to this force-directed layout, the position now of the nodes has a meaning, because the stuff that is close together because it's being pulled together by the edges, okay? Everyone cool with that? Okay. And once again, I mean, this is all work that was started by Eli in the lab. And of course, once you have built your graph, you can start overlaying information like you did, like I showed you previously with SPADE. And so, for instance, this is the exact same bone marrow data set from the science paper. Unfortunately, the colors are very poor with this projector. But if you start coloring by expression, you see that this region here is in CD3, this is CD4, and this is CD8. So essentially, this blob here are your CD4 T cells, and this blob here are your CD8 T cells. Then if you color by CD20, wow, that's horrible. But there is a blob here. There's a clump of nodes here that are your B cells. CD111, 123 show you that these are your PDCs. And so, I mean, it's very hard to see, but there are actually two close blobs here in the B cells. And so the expression of CD123 is telling you that these are the early cells and these are the mature B cells. Okay, so this is exactly the same that you would do with a SPADE tree, but now you're projecting the information on this graph. And the CD11B tells you that these are the monocytes. So once you have this representation, of course, there are a ton of algorithms that are called community detection algorithms that will essentially scan this graph and will find regions of nodes which have a very high density of connections, and we call that a population. So you can do this manually, as I'm doing it here, or you can also run one of these algorithms that will tell you that here there is a clump of cells, of clusters that are all close together, so probably they represent the same population. And the same approach can also be used to compare multiple samples. Like, for instance, this graph here is 21 different patient samples. And now each cluster, so all the patients have been clustered independently. So each node here contains cells from a single patient, okay? but then the clusters have been laid out together in the same graph. And here, like, they are colored by patient indication, by patient, essentially. So that all the stuff that has the same color comes from the same patient. And of course, I mean, you cannot see anything here, but then if you color, so there were two different subtype of tumors in this data set, and you can see that this population here, this group of clusters here, that is flying away from the rest, only belongs to one subtype of the tumor. So here now, instead of being colored by patient, they're colored green or red by tumor subtype. And you can see that this population is specific to a subtype of the tumor, okay? So this gives you, I mean, the whole concept of using force-directed layout and graphs gives you also a very powerful way to detect a group of clusters that are specific to a patient, to a condition, or to a sample.
And I just say that then you could go in and look at the markers that are expressed in the red cells versus the green to say what are the principal component differences between those cell types that you would not have perhaps seen previously.
Mm-hmm. Yeah. So the advantages of this whole effort is that the overall shape of the graph is the same for similar samples, which means that essentially, since now there are multiple connections for each node, even if your different biological replicate has a connectivity that is a little bit different, it will still end up in a similar shape. While the problem with SPADE is that since there is a single connectivity, essentially for the cluster, then minor variation will end up changing the shape very much. While here, in this graph, I mean, what I'm saying is essentially that even if this tiny little connection here is wrong, or that some of these connections are wrong, the overall shape, since there are more connections per nodes, this effect is averaged out so that the similar samples would give you similar graphs. The other thing, as I was discussing, that the layout is informative, so the whole point of this thing is to have an informative layout, so to have the position of the nodes that is also meaningful, while in SPADE only the connectivity is what matters. So this, I mean, after this, we wanted to more in-depth analyze and address the need of analyzing multiple samples. And that's something that I've developed, and a method to explicitly analyze to explicitly address the need for analyzing multiple samples. And we had a few goals in developing this thing, and one was actually to being able to integrate prior knowledge in the analysis, right? Because you can still take your set of data set, gate it manually, and you can still get a ton of information from it. I mean, you can still identify a lot of the stuff that you know about. And so it'd be nice to have a way to integrate this prior knowledge in your analysis without always pretending that you don't know anything about the data set. The other thing that we really wanted to do was have the possibility to independently cluster each sample, because this gives you a very useful tool, which is the possibility of mapping a new sample to a reference. So you run your 10 samples to do your whole analysis, and then 2 months later you run another 10 samples. And the idea is that you will process these samples independently and being able to map them on the same reference, so that you don't have to always redo your analysis, but you can add on to your existing analysis as your study progresses. And so, of course, this method has also to be able to display the variation between the new stuff you're mapping to the reference and the existing reference. So there is both an issue of consistency. You want similar things to be similar, but you also want the method to be able to show that there is variation in the new things that you have analyzed. And so this resulted in this development of this method that is very long acronym I just call scaffold for simplicity, and I want now show you how this works and the results that we got when applying it to a particular data set. So the idea is to build a map that explicitly incorporates prior knowledge. So now in your graph, there are two different kind of nodes. There are the clusters, which you have defined as previously, but then there are these landmark nodes or reference nodes that are represented as a red node here that represent your prior knowledge. Okay, so I know that this red node here represent my gated CD4 T cells, okay? And this other landmark red node here represent my gated CD8 T cells. So these red nodes are what's your prior knowledge, and this one, the blue nodes are the cluster that you have identified automatically from the data. Okay, and once again, the clusters are connected to the landmark with a weight that is proportional to their similarity. Okay, so for instance, this cell here is very close to the CD4 node, because this is as marker values that are very similar to those of a CD4 T cell. While, for instance, this cell here is sitting between CD4 and CD8 T cells, because the marker values of this particular cluster are in between the marker values of CD4 T cell and CD4 and CD8 T cells. Okay, so once again, the idea is to connect stuff with a weight that is proportional to the phenotypic similarity. And so this is something that turned out to be very useful with a data set that Matt Spitzer in the lab had been producing, which was like a gigantic effort. And essentially here we're dealing with 20 different mice in 10 different tissues. So it was looking at the immune cell in the spleen, bone marrow, blood, colon, lung, etc. So 10 different tissues of the mice analyzed with 39 surface markers. So a gigantic amount of data, 20 mice, 10 tissues, 39 markers per experiment. And the idea here was to use one sample specifically as a reference, which in this case is bone marrow. And the idea of using bone marrow is because we expect bone marrow to mostly contain all the cell types that are then found in the rest of the body. So the bone marrow sample was going to be the one that will act as a reference and will contain all the cell types that we're going to then see in all the other regions of the body. And so the first step of this analysis was to gate out, and this is something that was informed by Matt's prior knowledge, to gate out the reference population. So you identified these are your CD4 T cells, CD8 T cells. So using standard gating, you gate out a number of population. But then in parallel, you also do your clustering, where you don't tell anything to the computer, just identify group of cells that are similar together. You don't put any prior knowledge, okay? But these are two things are done in parallel. So on one side, you do the clustering. On the other side, on the same data, you do the gating. And so the reference gated population are what define the landmark nodes, which are the red nodes in the graph. So these are your known CD4 cell, CD8 cell, etc. And the unsupervised clustering define the blue nodes in the graph, which are the clusters which you don't know anything about. The only thing you know is that the stuff that's in the cluster is similar, okay? So now you connect all these things in a graph, like I was showing you before. And so essentially, you connect the clusters to the landmark nodes according to their phenotypic similarity. And this is done, this whole analysis is done for the bone marrow. Okay, so how do you bring in the additional sample? Well, what you do is now once you've built this initial map on bone marrow, what you do is you fix the position of the landmark nodes. So the position and the identity of the red nodes is always going to be the same, and it's fixed. Okay, so you maintain those. But then you go in and you cluster all the other samples independently. So you cluster your thymus, you cluster your blood, your spleen, blah, blah, blah. And you connect, for each tissue, you connect the clusters to the red nodes with the same criteria that you use here. So the weight is proportional to the phenotypic similarity. So what this means is that in all these maps, the position of the landmark nodes that provide the reference is always the same, but the way that the clusters are connected to these landmark nodes is different because the samples have been clustered independently. Okay? So I'm going to show you, after all this babbage, I'm going to show you what this looks like in practice. So this is the map that was built on bone marrow. So for instance, each red node here is a population. These are monocyte neutrophils. These are the CD4 T cells, CD8, NK, B cells, PDC, etc. Okay? And so the fact that all these blue nodes here are close to the B cell node means essentially these are B cells. Okay? And these blue nodes here that sit close to the CD8 are CD8 T cells, and so on and so forth. Okay? And the important thing is that the position of everything here is informative. So the fact it's not by chance that CD4 and CD8 T cells are close together. It's because they're similar in phenotypic space. Same for these other groups of cells, or these are all the progenitors. And so the position of the landmark nodes and the blue nodes is meaningful in this graph. And for instance, one thing, if you compare this with blood, so this is now the position of the red nodes is fixed, and we go in with blood. And you see, for instance, if I go back and forth between the two, you see that blood contains much more circulating T cells and doesn't contain all the progenitors that are only found in the marrow. Okay? So you see that all these nodes here, there is nothing connected to it in the blood. Okay? Because the blood does not contain those progenitors, which were in the bone marrow. Okay, this is bone marrow, blood. And the reason why you're able to make this visual comparison very quick and very effectively is the fact that the general structure of the graph is always the same. Okay, so the whole point of this exercise was to have a common reference that was always the same, on which you can overlay additional samples. And another thing that is important is that since now the position, as I was telling you, is meaning, so this is the thymus. Okay, so what does this mean? That there are all blue nodes here that are flying off in the distance from these red nodes here, okay? So this is another thing that is very important. And so the fact that this stuff is flying off from a landmark node doesn't mean that there is a mistake or an error in the data, anything like that. It just means that these cells here have something in common with CD8 or CD4 T cells, but they are not quite right, okay? And so the difference that is here is in the thymus, you get also double positive population. So these nodes that are flying off here are actually CD8 and CD4 developing T cells that are only found in the thymus, okay? So by seeing the stuff that is divergent, we can go in and look at what's particular to that sample, okay? So you have this sample here, you see that stuff is flying off, you go in and you see what is peculiar about those cells, okay? So in this case, for instance, that's CD4 and CD8 T cells. And I mean, of course, this nicely integrates with all the stuff that I was talking about force direct layout, because if you want, you can go in and zoom specifically on the T cells. And if you do this now is an unsupervised layout, so going back to where we were before, where we don't put any prior knowledge, but we can see that so now the graph is colored by tissue, and you can see that this blob here that is very distinct from all the rest, and is the CD4, CD8 double positive, is only found in the thymus. I'm sorry, the coloring here is not very good on this projector, but this population here is specific to the thymus. So by using the force-directed layout graph, even in an unsupervised mode, you can see that that population is specific to the thymus. And the other thing which we are now working on is the idea of mapping additional archival data on this reference map that we've built. And so, for instance, this is the original bone marrow side of data. And this is an experiment from a different mice done by fluorescence. So this is actually fluorescence data mapped on side of data, which is kind of diciamo, a tall comparison to make. But essentially, what's happening here is that, of course, the layout is not as clear and as nice as the side of one, but you can still call out the major difference. And for instance, the fact that there is a lot of aggregation here and a blob, an indistinct blob here, is not due to the fact that this is fluorescence and this is side off, but it's due to the fact that we're only using 8 markers. So of course the resolution is going to be lower if you just use 8 markers instead of 39. But actually, if you take the original side of data and you use only the same 8 markers, you actually get a picture that is very similar. So the fact that the fluorescence map look a little bit worse than the original side of map is not due to the fact that it's fluorescence versus side of, but due to the fact that it's 8 markers versus 39 markers. And the other thing we're trying to do is mapping, trying to map even across organisms. So taking a mouse sample and the human sample, and essentially establishing which markers are equivalent between human and mouse, and doing the same thing. And I mean, one thing that is super apparent in this graph is that, which is what was known, is that mouse have a lot more circulating B cells in the blood. So mouse B cell, like you can see that there is a lot of stuff aggregated around the B cell node in the mouse blood, and there is not that much stuff in the human blood, because it was known that mice contain more B cells in the blood. So to give you a quick recap of the difference between spade and scaffold, so first of all, the samples are clustered independently. That's the most important thing. And which means that additional sample can be added to your analysis without having to rebuild the structure. The other very important difference is that we explicitly incorporate prior knowledge. So we're not throwing away our knowledge about gating. We're using gating to inform the construction of the map. And data is viewed with respect to a reference. So you are projecting everything with respect to the reference that you know about. Okay, so that's a very important point. You're building your structure on the reference, and then you're using that structure to view the rest of the data. It's more robust, okay? And also, I mean, as I was showing you, it allows mapping of archival data, and this is something that can still be perfect, and it's something that we are currently working on at the moment. So, I mean, after all this method talk, I just want to have a recap slide about the general coordinates that define your experiment usually. Of course, I mean, you can always find exception, but I want to give you an overall framework for how to think about your experiment. So first of all, you have your markers that define a very important coordinate of your experiment. So you have functional and phenotypic markers, okay? And we've already discussed about the distinction. And this is going to be like one of the axes of your experiment, which markers you're measuring. Then, of course, you're going to have different experimental conditions. You're going to have your beta, your different stimuli, and all of that, okay? And another axis is going to be your individuals. You will have measured markers in different stimulation conditions in different individuals. So this is like a 3D space. And so these are the pieces of data. So for the first individual, you have measured in the basal, you will measure phenotypic and functional markers, and then you will measure stimuli 1, phenotypic and functional, and all of that. And each one of these things is going to be an FCS file, so a site of run. In a single site of run, you will measure for your basal condition, your functional phenotypic, for the stim1 functional phenotypic, and so on and so forth. And we, in general, assume that this stuff is invariant. So that in the most basic experiment, you will assume that the population you're looking at are always more or less the same, and so you will use this to define the structure. Of course, it's very possible that your disease will have an enlarged compartment or a compartment that shrinks, but you will assume that this is more or less what it's more constant unless than the functional markers, because the functional markers are what really varies across all your different conditions, and so it's the stuff that you want to overlay on the basal to color your tree, color your graph, and see what cells are responding to your different cytokines or whatnot. And of course, I mean, you might have these for different individuals, so this provides another axis to your experiment, all the different individuals that you've measured. And as I was telling you, the main difference is that with SPADE, you have to analyze all this stuff together, while with this other meta scaffold, you can analyze things independently. So I'm now down to the acknowledgement, of course, Gary. And so the work on force-directed layout in the lab was started by Eli. The data for the mice experiment was generated by Matt Spitzer, and also Karashan helped with this. And SPADE was the work of Peng Q in Silvia Perviti's lab, and of Erin Simmons. And of course, thanks to all the rest of the lab for discussions and input. And I'll be happy to take your questions. Yeah.
You mentioned that with the SPADE, every time you ran the same sample repetitively, you get a different tree.
Yeah. So.
Does that happen with the scaffold other than the?
So the thing, the overall shape, I think, is going to be more consistent. I mean, as what Greg was telling you is true. I mean, it's still gonna have the same qualitative feature, the tree that you get from one run to the other. But I think it's gonna be more difficult to recognize the shape. That the shape is similar because the position of the nodes is not informative. While with this other method, I think it will be easier to see that the overall shape that you're getting is more similar. Okay, which doesn't mean that. I mean, doesn't mean that the SPADE tree is, doesn't have any meaning because it's different every time. It's just that since the position is not informative, it's more difficult for you to see the relationship between the different trees. Okay. Yep.
So do you generate the force-directed layouts right in SPADE, or is it?
No, this is something.
Completely separate?
Yeah.
When you're choosing those landmark populations. In Matt Spitzer's example with the different mouse tissues, you use bone marrow as a reference, but you could have probably overlaid all of the different tissue types, right, in a synthetic data set and chosen every possible cell type.
Yeah.
If you ran into a situation with thymus where there was a cell type that wasn't present in bone marrow, the double positive T cells, right?
Yeah. Okay. So if we had
you had overlaid everything.
Yeah, absolutely. So if you do these things this way, then it's never going to happen that there is something like what was happening with thymus. But I mean, the idea was to show you that this thing can be done in a more simple way by just taking annotating one data set and then projecting stuff on top. But then, of course, I mean, you're perfectly right. If we had gated everything from the beginning and defined landmark nodes for everything, then we wouldn't have had this observation in the thymus.
And what do you do when you have a population that I guess you would never have predicted? I guess it would be kind of similar to the thymus situation where
Yeah.
the cells are just
Okay.
everywhere.
So actually, I mean, this happened in this data set. I mean, so there was a data set, like Matt was able to identify a novel population from this stuff by looking at stuff that was flying off of one specific node in one specific tissue. So that's something that has happened. So the way that this stuff show, like when you have something that is really completely different from what you have in your landmark nodes, it's going to be identified by something that is connected to something but is flying off very much in the distance, and this has happened, and he has identified something novel, which I think was pretty exciting.
What happens if your staining is a little imperfect or some of your markers aren't working? How do you
Okay.
address that?
So the thing, I mean, clearly depends on having data where the quality is more or less consistent. But I must say that, I mean, I was incredibly surprised by how well the fluorescent data map on the side of data. So I would say that once you normalize the data, because when you put everything on the log scale, essentially a lot of these differences are really compressed. And so I would say that even if there is variation, it's still possible to do it. But then, of course, I mean, if you tell me I have this data set which has completely crazy marker values that are completely different from my reference, then of course it's not going to work. I mean, this still relies on the fact that the numbers must be comparable between different samples.
And if one particular marker just falls out in one sample, I guess it's unlikely to
I don't think it's going to be that much of a problem. I mean, it always, because the distance is calculated in 40 dimensions, so it all depends on what's the end weight of that marker on the distance that you're calculating.